as much as we would love to be talking about other things, the number of important developments in the land of COVID is so many, and the amount that rides not only on our ongoing developing understanding of the way COVID and COVID treatments work, but also on our ability to go back and figure out what we got right and what we did not have right in the past. Now, this week, this episode, we are going to talk about a result that has recently emerged in which the two major types of vaccines for COVID, that is to say the adenovectored vaccines and the mRNA lipid nanoparticle vectored vaccines, were assessed with the measure of all-cause mortality. Mm-hmm. Now, that is an important thing to do, and the reason that it is an important thing to do is that vaccines, as we have pointed out hundreds of times maybe, are basically an intervention into a series of complex systems. That series of complex systems being at least three-layered. You've got the immune system, which is a complex system, inside of the critter, that is the human, which is a complex system, inside a society of people in which a pathogen produces an epidemic. So all of these complex systems are going to be affected by the various interventions that we might seek to deploy, and their net effect is what we ought to be interested in. So this recent study, which is technically not peer-reviewed yet, which means that it was not handed to anonymous reviewers and their comments then delivered back to the editors, and the editors then deciding to publish it. But what has been peer-reviewed, in fact, by Martin Kulldorff and colleagues who looked through and reported on this. Zach, do you want to show the website that I sent you? Martin wrote about these vaccines. In any case, so as you and I, Heather, have been talking about from the beginning of the pandemic, peer review does not mean review by peers. Peer review is I would argue, a kind of jargon that obscures the reality in which peers are anonymously given a paper to review by editors, and the amount of skullduggery that flies beneath the radar under this banner of peer review is great. And while some of us oppose this kind of peer review, none of us who are worth our salt as scientists oppose the idea of peers reviewing work, but we think it would be much better done out in the open, transparently. Yes. I'm
wondering if before you show this article by Martin Kulldorff, you want to just show the actual preprint here.
Yes, I think we need to show the preprint, which I also sent you,
I've got on my screen as well.
You've got it on your screen too.
Isn't this the paper?
That is absolutely the paper. So either way. So here we have it. And
it's called, for those just listening, Viewpoint, Randomized Clinical Trials of COVID-19 Vaccines. Do adenovirus-vector vaccines have beneficial non-specific effects? And just as a reminder, although most listeners and viewers will already recognize this, we have two main classes of vaccines that at least in the weird world have been widely available: the mRNA vaccines produced by Pfizer and Moderna, two different ones, and the adenovirus-vectored vaccines produced by Johnson & Johnson, which is the only adenovirus-vectored vaccine that's been allowed in the United States, and then also AstraZeneca.
Right, and so actually maybe we should just do a little of the biology here. Both of these kinds of vaccines are novel. And the miracle, which is their rapid production in the aftermath of COVID, also presents us with a problem, which is that we've got two platforms, essentially, in which a vaccine can be manufactured. Now, apparently, it can be done very, very quickly, that we know very little about. We just don't know what their effects are. And they work in two, I would argue, radically different ways. One of them takes an adenovirus and basically implants a antigen, or the message that produces an antigen, into this virus, which is capable of infecting human cells. And the other one takes an mRNA, which is a message that a cell natively transcribes, and introduces it into the cell as if it had just come from the nucleus. And the cell then faithfully transcribes and actually translates this message into a protein, which is supposed to then lodge in the cell surface. But in the mRNA case, instead of being delivered by a virus, which has evolved to infect human cells, it is delivered by what's effectively a pseudovirus, which is a coat of these lipid nanoparticles. And these lipid nanoparticles, lipid means fat, and the lipid nanoparticles, because they are composed of fat and because the surface of all cells is made of a fat-based membrane, causes it to fuse, like dissolves like, and basically the lipid nanoparticles touch a cell, the message is spilled in, and then the ribosomes in the cytoplasm translate the message into a protein, which then gets displayed on the surface of the cell, hopefully, and the immune system learns to recognize it. Now, you and I have talked at length about the hazard of these new technologies, especially the lipid nanoparticle one. And the reason that the lipid nanoparticle one has caught our attention as something particularly concerning is that there is no specificity whatsoever in where these lipid nanoparticles deliver their message. They will deliver it to whatever cells are most hospitable, and there is no reason to think that the cells that are most usefully delivered this message are the ones that will pick it up. It will circulate, in the form that the manufacturers intended it. It's supposed to stay effectively in the arm where it's injected, and it's supposed to be those cells, presumably the muscle cells, which pick it up.
Right. And a critical distinction being that whereas with traditional vaccine technology, of which there are also two types, and one of which is more ancient than the other, as you'll talk about, but attenuated, but still live versus dead vaccines. In both of those cases, the idea is we're going to put something in your body, the goal of which is to get the attention of your immune system so that it can learn how to deal with such a pathogen if it runs into it in its wild type. And in this case, it is quite not that. So it is lacking specificity, and the expectation is not that in its form in which it is injected, the immune system will recognize it, although that might also be the case, but that it will move into cells and produce copies of, for the most part, the spike protein. And can I just read a couple sentences from the beginning of the introduction here of this?
By the way, do you want to say who the authors are?
Yes, the authors are, I'm going to butcher most of the names here, Christine Benn, Frederick Schultz-Buchholzer, Sebastian Nielsen, Mihai Netea, I apologize, and Peter Abbey. This, again, is the preprint called Randomized Clinical Trials of COVID-19 Vaccines. Do Adenovirus-Vector Vaccines Have Beneficial Nonspecific Effects? I want to read just a couple sentences from the intro and then sort of the conclusion out of the abstract, even though that's not usually the order in which one shares stuff. Introduction. Within the current understanding of vaccines, it is logical to assume that COVID-19 vaccines reduce overall mortality, corresponding to the number of COVID-19 deaths prevented. Okay? However, there is now ample evidence that vaccines can have broad heterologous effects on the immune system. These effects can lead to additional protection or increasing susceptibility to unrelated infections or even other non-infectious immune-mediated diseases. Therefore, as it has now been established in numerous studies, vaccines may have completely unexpected effects on overall mortality, different from what could be anticipated based on the protection against the vaccine-targeted disease. That is an excellent précis right there of basically the argument that then the first author on the paper, Christine Benn, makes in an interview with Unheard that you will also talk about, that she did this week, for why, unlike what was done, for instance, in the trials that Pfizer ran, we should not be focusing only on effect on that disease, but overall mortality, all-cause mortality, to use the term of art, right? So and frankly, it's easier. It's easier to look at all-cause mortality because you don't have to end up dithering, and dithering minimizes the importance of this. But as many of us have been trying to figure out these last 2 years, and for many people, for much longer than that, like, well, in the case of COVID, it's only the last 2 years, 2+ now, did she die of COVID or with COVID? And because, especially now with Omicron being so highly transmissible and so many people have had it or still have evidence of it circulating, it is quite clear, quite a number of, sometimes presumably unintentional, sometimes absolutely intentional, messing with the numbers such that all of these deaths that are attributed to COVID are not actually deaths that happened because of COVID, but deaths that happened while the person also was infected with COVID. People who entered hospital for entirely other reasons were then tested, found to have COVID, died for those entirely other reasons, and were listed as having died of COVID. So all of that problem is avoided if you focus on all-cause mortality. In those people who got, say, mRNA vaccines versus adenovirus-vector vaccines, and that's the comparison being made here, not to unvaccinated people, was all-cause mortality, or
that's not exactly right.
No, it's not. It's not exactly right. So, in these three groups, is all-cause mortality, better or worse? And the abstract reveals here.
Can we hold off? Can we lay a little more groundwork?
Okay, then he shouldn't be showing the screen.
Yeah, I mean,
which has the answer on it.
It's not a secret, but
okay.
First of all, this comes in the context, right? And you hinted at that context, but I think that context, you have to understand it in order to see why a study like this is likely to tell you something really important that you might not know. So, Dr. Ben talks about prior work, which has revealed a pattern. I believe that she and her team studied 10 vaccines that are just part of the standard schedule of vaccines for children. Mm-hmm. And the pattern that they found was that the live attenuated virus vaccines tended to increase longevity. They decreased all-cause mortality, and the all-cause mortality decrease could not be attributed to the target disease entirely itself.
Could not entirely be attributed to that.
It's right. So, for example, in the case of the measles vaccine, there was a benefit in the reduction of measles, which did account for some fraction of the reduction in overall loss of life, but it didn't account for all of it, not even close. So the point is, there are non-specific effects of these vaccines, and we don't entirely understand what they come from. Mm-hmm. One could imagine, for example, this is one plausible example, that cross-reactivity, because viruses are related to each other, being vaccinated against one virus might create immunity to another virus that had similar surface structure. And
for which maybe we're not as familiar. We don't have a name yet. We certainly don't have a vaccine, but now you've got some baseline immunity to something that the medical establishment isn't formally looking out for.
Right. And it doesn't have to be that direct. It could also be that because the immune system exerts a selective pressure on the viruses themselves, viruses that aren't closely related may come to resemble each other because what they're doing is they are evolving to be invisible to the immune system. And so cross-reactivity doesn't even require that the viruses be related. But in any case, the basic point is if you look at the attenuated virus vaccines, what you see is an all-cause mortality benefit. You get the exact opposite when you look at the killed virus vaccines.
So again, these are the two broad types of vaccines that existed before, a couple of years ago.
Right. Now,
and what's an example of a killed virus vaccine that we will most of us have gotten?
I am not going to take. Well, let's see if we can deduce.
I just don't.
We can deduce it from the fact of needing boosters. But in any case, I'm not going to guess. But the point is, when people who are DarkHorse listeners will have heard the following logic discussed by us here before. If you have an attenuated virus, a live virus vaccine, do you have an answer?
This is just this is off of a site called Passport Health Global, so I'm not going to vouch for it beyond that. But some attenuated live virus vaccines include MMR, as you said, yellow fever, oral polio vaccination, and oral typhoid. And some dead vaccines include, they say polio here, and they're going to mean not oral, cholera, cholera is a crap vaccine long before any of this. It's just got really low efficacy. Influenza, Hep A, and typhoid. And again, it's the non-oral typhoid. Yeah.
So the logic works something like this. When you get an attenuated virus vaccine, that is a living vaccine that is a weakened form, it doesn't make you sick, and hopefully it can't transmit from one person to another. right? All bets are off if something figures out how to jump from one individual to another. But a attenuated virus that can infect cells enough to give you a very mild infection, hopefully one that you can't even detect you've got anything. Maybe you've got, your arm has a slight fever or something like that.
Fatigued for a day, this sort of thing.
Yeah. That, because it is an infection, right, causes the immune system to have the right reaction, right? They work very well for this reason. They're also a little scary for this reason because you're dealing with an organism that has heredity and therefore could evolve. It doesn't evolve very well if it doesn't move from one person to the next. But as soon as that trick gets introduced, it's a very different thing. Yep. But in the case of a killed virus or a antigen from a killed virus, a piece of a virus that you're delivering to the immune system may spot it because it's foreign, but it doesn't look like an infection. And so the reaction is often very weak. So two things are true. One, you may get more than one vaccination in order to introduce the same antigen multiple times, which sort of simulates an infection. And two,
trigger is the goal here, unlike in much of social space now. Like you're trying to trigger your immune system.
Trying to trigger the immune system.
What is this? Right?
But then the thing that you and I have talked about, hey, wait a minute, what the heck is this technology? And whoever said that was safe is the adjuvants. And the adjuvants are basically immune irritants that cause the immune system to react like there's an infection, which then raises all kinds of questions about how good are they at spotting the thing that the vaccine manufacturer wants them to react to versus the stuff that was involved in the food that you ate or your own tissues, right? In other words, annoying your immune system with an adjuvant is basically an invitation to the inappropriate activity of the immune system, either in the form of allergies or autoimmunity. And so anyway, what Dr. Ben reveals in her interview about this paper, which is narrower, is that in this prior work, the answer came out pretty clear. The dead virus or pieces of virus, the antigen-based adjuvant-containing vaccines reduced longevity, increased all-cause mortality, and the live virus vaccines decreased all-cause mortality. So
just are live, also known as attenuated virus vaccines, inherently adjuvant-free? Do you know the answer to this?
So.
Okay.
I know that's the general pattern. It may be 100%, but in any case, so they
have all sorts of things going for them.
Right.
Yeah.
So,
and I mean, this just reminds me, and this is what we've been saying from the beginning of COVID and the prospect of vaccines. Like, oh, goody, cool, vaccines. Vaccines are one of, broadly speaking, the three major successes of Western medicine, antibiotics and surgery being the other two. And, oh, by the way, just because we call it a vaccine doesn't mean it's awesome. And especially if we are in an environment in which people are willing to play all sorts of games with words and slot anything over into vaccine space, and at the same time start redefining what vaccine means and what vaccination means and what anti-vaxxer means, well, there's just no ability to communicate anything clearly. And so really it shouldn't be necessary for all sorts of people who aren't inherently biologists or don't want to be paying attention to this sort of thing to keep track of all the different types of things that are currently being called vaccines. But for those of us who are dealing with both, as everyone is, a global crisis, some of which is based in health and some of which is based in politics, and are trying to be clear and also don't simply have the position either that no vaccines are good, I want to stay away from all of them, or anything that you choose to call a vaccine is good and therefore shoot me up. That vast middle ground is actually the scientifically rigorous place to be, and therefore we feel the need to talk through why both the mRNA and the adenovirus-vector vaccines that were introduced, I think the adenovirus-vector vaccines were also introduced with COVID, or did they pre-exist COVID?
I can't say what's been done in other creatures, but
for humans,
this is the first time for humans.
I believe this is right too, and certainly for mRNA. I mean, they were trying some stuff, but it wasn't working. But with regard to the two, and I don't know if we're missing other categories, but so far we've talked about four sort of categories of vaccines here today, and the two categories of vaccines that pre-existed, that existed 5 years ago, being dead or parts of viruses or antigen. You called them antigen vaccines. Is and I'm not sure I've heard that, but it makes sense. Versus live, also known as attenuated vaccines. Of all of these, there is one and only one, and it is the original one. Right, it is the original formulation of vaccines, I believe, right? The attenuated or live vaccines that actually appear, given what Dr. Ben's past research has indicated, and what, frankly, well more than a century of research has indicated, to reduce all-cause mortality in those who receive them.
Right. Now, I will say the work on the live attenuated versus the killed virus vaccines and their relatives, I didn't know that work existed. I didn't know that anybody had done all-cause mortality work. And so it's not purely independent. But I will point out, this is reflective of exactly the logic that you and I worked our way towards. We figured this out. This is what we predicted. Yep. Now, there's one aspect of this that I didn't predict, which is worth pointing out, too, because the question is, how good's the model?
Yeah.
Right? I didn't predict a all-cause mortality benefit from generalized administration of a live virus vaccine. Okay, that's a little surprising to me, right?
That goes beyond
the target disease.
Yeah. So, and that wasn't included in what you said. So there's all-cause mortality that may increase because the benefit against that disease is so high.
Yes.
But all-cause mortality that is not fully explicable by the benefit in fighting the target disease.
Right. Whereas,
terrific. Yep.
There are going to be other effects. You're intervening in a layered series of complex systems with a therapy that's going to have multiple impacts, almost all of them negative in the case of the killed virus adjuvant vaccines.
Right.
That we did say. Right, that did seem very likely, and turns out to be true.